Hello everyone, am I audible and clearly visible to all of you? Hello everyone, welcome to Unacademy NEET English channel. Myself Gopika, your biology educator. Okay, we are back again with one session of biological classification part 2, right? Last time we did part 1 and now we are doing part 2. Okay, yes, good evening. How are you all doing today? How are you all feeling today? Good evening. How are you all today? Are you all fine? Are you all ready for the class? Did you revise part 1? So I will be asking questions, right? I'll be asking questions. Yes. Okay. Okay. Fine. Okay, great. Okay, most of you have revised. Okay, we will see. I'll be asking a lot of questions and I want all of you to have your notebook, pen, right? Wherever you made part 1 notes, I want you to continue. I don't want you to write in one book and then later try to search for that. Okay, so carry the same notebook throughout. Okay, so that by the end you will not find it difficult to go search for all your notes, right? Okay, so students, if you haven't shared the link with your friends, quickly share the link and ask all your friends to join so many of you can get benefited with the chapter, because you know this chapter is complicated and some of you might find it easy, some of you might find it difficult. Okay, so make sure that you share the link with your friends so that everyone is helped, right? Okay, so quickly do that, share the link and ask your friends to join. And if you haven't joined any batch, okay, if you haven't taken any course or you're still thinking what should I do after 12th, right, you should join Unacademy's course and use my code, that is Gopika, Gopika. Okay, do not forget, no matter you're taking UPSC, no matter you're taking any other, competitive exams, you can still use my code and get maximum discounts. Okay, and do not forget to join my special class, because I saw many of you have commented on my previous video that ma'am, biomolecules chapter is really difficult. I started special class series for biomolecules, so do not forget, go watch. Part 1 is already live. Okay, yesterday I did part 1. So today I'll be doing part 2 at 8 o'clock. So do not forget to join. Whoever is finding biomolecules chapter difficult, whoever put it in the comment section, I have already started it in the special class for you people. Okay, so do not forget to join. My link is in the description box. Okay, who is finding it difficult, how to join special class, who is confused, who is first to this platform, everything is in the description box. Fine. Okay, being said that, before I start the chapter, there was one doubt that was being asked in the comments. Okay, and I think that doubt is there in most of you. Okay, the doubt is nothing but Chlamydomonas and Chlorella, why are we studying it under algae even though it is put under kingdom Protista according to Whittaker? Students, do understand, according to Whittaker did not care about if it's going to do photosynthesis or not. Okay, he did not care about that. He cared about the cell wall, cell structure. Okay, so they were put under Protista because he found them, okay, they are eukaryotic and they are unicellular. Okay, but according to the latest classification, people have understood that it is better that Chlamydomonas and Chlorella comes under algae, right, under plant, because they are having the ability to do photosynthesis and they also have pigments. Okay, so if the question comes in NEET, according to Whittaker, Chlamydomonas should be in which? That answer should be kingdom Protista. Okay, kingdom Protista. So your what is that, your textbook plant kingdom is not according to Whittaker's, but is according to the latest classification. Clear? The doubt is clear. Some of you had asked it in my last video comments. I do read your comments, so if you have any doubts, do not hesitate drop the comment. I will be taking it in the next class. Clear? I hope that doubt is clear, okay? So, in case the question comes according to Whittaker, where is Chlamydomonas coming? You have to put kingdom Protista, okay? But according to the latest classification, okay, presence of cell wall, all of this has made them, classified. We study it under algae because that makes more sense. Schematic representation or the way to study is easier when it comes under kingdom Plantae. Yes, great. Okay, so let's start the session. So, if you are all ready, use fire symbol in the chat box and let me know, ma'am, we are ready to start the session. And you should be ready with your notebooks as well, okay? Use the fire symbol and let me know that we are ready for the session. Quickly. Yes, ready? Yes, okay. Why there is no energy? What happened? What happened? All are sleeping. All are sleeping. Quickly send fire symbols in the chat box so I can start the session. Yes, great. Okay, fine. Students, so last class we learned up to Archaebacteria. I told you that we will start with Eubacteria this class, okay? I will be asking questions. Okay? As of now, we will start the class. So, under kingdom Monera, we came across Archaebacteria and Eubacteria, right? So, now look here. Eubacteria is also called as true bacteria, okay? 1 minute. It's also called as true bacteria. Why this is called true bacteria? Because when we studied Archaebacteria, we saw that the cell wall of Archaebacteria was made up of pseudomurine, right? False cell wall, right? It was just to protect Archaebacteria from other lysozymes which are secreted by host immune system. But when it comes to Eubacteria, they have a proper cell wall which is made up of peptidoglycan, okay? You know peptidoglycan is very rigid, okay? When we studied kingdom Monera, I happen to tell you how strong peptidoglycan is. That is the reason why bacteria is able to withstand any extreme condition. So, here in Eubacteria, it is called as true bacteria because of the presence of peptidoglycan, okay? Glycan on their cell wall, right? Cell wall is made up of peptidoglycan. Fine, okay? Now, they have a presence of rigid cell wall. This is actually a common characteristics of kingdom Monera. And then they have flagellum and they are motile. So, if they have flagella, that means they are definitely motile, right? Now, Eubacteria has two types. Now, students, listen carefully. Eubacteria has two types, okay? Mycoplasma and cyanobacteria. This is one of the favorite questions of NEET, okay? Because they have lot of exceptional characters. So, this is why, this question directly is asked in NEET. It will not be, very complicatedly asked. It will be directly asked. Clear? Clear? So, Eubacteria is divided into cyanobacteria. Cyanobacteria other name. What is the cyanobacteria other name? What is cyanobacteria's other name? BGA. What is it? BGA? Blue-green algae. Now, do you see here blue-green algae? Now, algae has come under kingdom Monera and blue-green algae means, okay, blue-green algae means what? It will do photosynthesis, right? It will do photosynthesis. No, under Plantae itself, you will study algae, okay? Under Plantae itself, you are going to study algae. You are not going to study algae as a separate kingdom. Got it? You will study it under Plantae only. For the flow, for the understanding, they have put Chlamydomonas and Chlorella under algae. I think your doubt is cleared, okay? So, cyanobacteria is also called as BGA, that is your blue-green algae, and then we have Mycoplasma, okay? Now, look here. This all about cyanobacteria, okay? Cyanobacteria is a unicellular, colonial and filamentous. Now, I will tell you what happens. Look here. So, your cyanobacteria, this color, okay? Your cyanobacteria is also called as BGA, right? They have the ability to form colonies. Okay. So, now what they are going to do? They are unicellular, okay. they'll form colonies like this, right? They'll form colonies which will look filamentous or thread-like. Okay, they will look filamentous or thread-like. Fine. Now, what is going to happen? To protect this layer, they are going to have, okay, they are going to have a mucilaginous sheath. Okay, they will be surrounded by a mucilaginous, jelly-like. You can see all these terms. Okay, you can use all this term. See, a jelly-like covering, okay, or a mucilaginous covering according to your NCERT. Okay, mucilaginous covering to protect them. Okay. Now, this is actually what? This is nostoc. Okay, the example is nostoc. This is a cyanobacteria. They can form colonies. Okay, they can form colonies. They will not stay individually, they will form colonies. Okay, and this colonies will look like a filament. Got it? Got it? Now, blue-green algae. What are the colors that you hear here? Blue-green algae. According to what we know that kingdom Monera, they can do autotrophic, chemosynthetic and all that, right? Now, you are going to see how they are going to do chemosynthetic. Now, in this, in this nostoc, there will be some specialized cell, okay, some specialized cell, the ones that I am drawing in red, no? Now, what are these people going to do? These cells are called heterocyst. What are these cells called? Heterocyst. These are specialized cells which are only seen in cyanobacteria. Okay, these heterocyst cells are seen in cyanobacteria. They help in nitrogen fixation. Nitrogen fixation. What do? They take the atmospheric nitrogen and convert it into ammonia. Okay, so this heterocyst is a specialized character of blue-green algae. Blue-green algae. Got it? So, they take atmospheric nitrogen and convert it. Okay, so that is why we call the cells as nitrogen fixing cells. Got it, students? Got the exceptions? Okay. Now, mucilaginous, heterocyst. Now, cyanobacteria, we will see what. Okay. Wait 1 minute. Yeah, we'll see here. Okay. Now, cyanobacteria is unicellular, colonial and filamentous. Now, you understood all these 3 points? All these 3 points you understood, right? Now, the colonies are generally surrounded by a gelatinous sheath, mucilaginous sheath, jelly-like, whatever you remember. Okay, whatever you remember. Okay. Cyanobacteria lacks a membrane-bound organelles. It is understood they are very small, they are primitive, they will not have all the organelles. Okay, they will not have all the organelles. Clear? Clear? Yes. Now, they have special cells called heterocyst. Now, these are the heterocyst cells that is shown in your NCERT, and this is a diagram from your NCERT. Okay, I haven't taken diagrams from outside because it will be confusing. You might see this, you might think about it, you might get worried. So, this is heterocyst nitrogen fixing cells. Nitrogen fixing cells. Okay, those who joined late, we are just doing eubacteria. Okay, we are doing eubacteria. Under eubacteria, you have mycoplasma and we have mycoplasma and cyanobacteria or your blue-green algae. Okay, blue-green algae. So, we are doing characteristics of your cyanobacteria, general characteristics, specialized characteristics, everything. Okay, fine. Example is nostoc. Now, let's see next one. Okay, now look here. They are non-motile, that means flagella is not there, flagella is completely absent. They can be found in marine, freshwater and terrestrial. Now, students, as I told you last class, even if they are present in terrestrial, will they be present everywhere? They will not be present everywhere, they will be only present in damp soil or in moist condition. Okay, they will be present in moist conditions. They cannot survive in dry conditions, right? They cannot survive in dry conditions. Now, this is one important thing which is not there in your NCERT but will be asked. Okay, so make a note. Okay. Hi Surya. Okay, gas vacuoles are found in all the water blooming. So understand, gas vacuoles are specialized vacuoles that is present in aquatic organisms. Okay, that means aquatic algae. Okay, this gas vacuoles, what do? They allow it to float or they allow it to have the buoyancy. Okay, now why should they have buoyancy? Why should they have buoyancy? Understand, students, when they float, okay, when they come up, they will get the oxygen and require light for them to do photosynthesis. Do you agree? Without light, can they do photosynthesis? They cannot do photosynthesis, right? So, this buoyancy, this gas vacuole allows them to float in whatever depth they want, so that they will get amplified, like enough amount of oxygen and sunlight, okay? So, this is the function of this. They will be able to get optimal sunlight and oxygen, okay? Oxygen for survival. Gas vacuole will do the job of buoyancy, that will allow them to have optimal sunlight and oxygen. Did you all write it down in your textbook? It is not there in your textbook. So, please write it down in your textbook right away. Okay, later you are not going to do. So, do it now. Okay. So, this gas vacuole is present in especially the ones which are aquatic. That means they live in water. Okay, they live in water. Okay. Fine. Yeah. So, they are photosynthetic from the name itself. BGA, blue green algae. What do you expect? They will definitely do photosynthesis. Agreed? Agreed? Right? Chemosynthetic autotrophic bacteria oxidizes various inorganic substances like nitrates, nitrites and ammonia. Here is where heterocyst is going to do the job. Okay? Heterocyst will do the job. Okay? Got it? Got it? Okay, understood students so far? Yes. Now, some examples are nostoc, anabina, spirulina, okay, spirulina and then oscillatoria. Now, spirulina you all know that it is used, okay, right, you know it is used by people who, the scientists who go to space, because this is very rich in protein, okay, this is very rich in protein. So, because they cannot have, you know, other food, they will take this in the form of tablets and they will use it in when they are going for any kind of discoveries, okay. Spirulina, then you have oscillatoria. Actually, oscillatoria you people would have studied it in your practicals, first year students would have studied it in your practicals, right? Oscillatoria. Understand that oscillatoria does not have heterocyst, okay. Still findings have been happening about oscillatoria having the presence of heterocyst in oscillatoria. Clear? Okay, do not make that confusion. Got it? Yes. So, can we quickly revise your cyanobacteria so far? Okay, so far what and all did we discuss? I am only going to tell you the words, right, what is important. So, cyanobacteria, we discussed that they are capable of forming colonies, which we haven't seen anywhere yet, right, formation of colonies, and that colonies are covered by mucilaginous sheath, right. Are you all with me? Are you all with me? Are you understanding? Yes. Mucilaginous sheath, right. Then we studied about special cells that are capable of doing nitrogen fixation. Those cells are called heterocyst, right. And then what did we study? They have gas vacuoles, okay, they have gas vacuoles that will help them with buoyancy, that will give them buoyancy, that will give them, okay, optimal amount of sunlight and oxygen. I want you all to write this. These are the exceptions that we have studied so far. Okay, these are the exceptions. Yes, are you all with me? Did you write so far? Did you understand? Tell me quickly in the chat box. Are you understanding? Okay, these are the exceptions. Okay, we'll proceed. Okay, now cyanobacteria, okay, cyanobacteria has few pigments, right. From the name itself, you know, BGA, it should have some pigments. Now, here is when you're going to see, okay, here is when you're going to see chlorophyll A, okay, chlorophyll A, right. chlorophyll A, which will give green color to the bacteria. Then we have phycocyanin that will give blue color, and phycoerythrin that will give red color to the bacteria. Okay. So now you see that cyanobacteria has so many colors, right? So many pigments, correct? This green is going to allow it to do photosynthesis, right? Photosynthesis. Okay, photosynthesis will be done because of chlorophyll A. Now, a lot of ponds turn green. Students, how many of you have seen when you go, through the village side, there will be ponds which will be not used a lot of times, right? Not used. You can see that there will be a completely green layer over it. How many of you have seen? How many of you have seen a completely green layer? Now, what is that green layer? That green layer is called your algal bloom. That means your algae has completely undergone rapid multiplication and covered the pond. Now, covered the pond. Now, you might be thinking, what is there in that? What is there in that? Students, do understand. Yes, good. Do understand that when such algal blooms cover the top of a pond, no, other aquatic organisms are going to end up dying because there is no sunlight that will be allowed inside. The oxygen, dissolved oxygen in the pond also will be completely used up by the algae for rapid multiplication. Do you get it? So that is a disadvantage of algal bloom. Other aquatic organisms will end up dying, and because of it they will not, the other like plants which are living, they will not get enough sunlight from because of this covered algal bloom. Got it? Now you can see that this is done because of cyanobacteria. Cyanobacteria can produce algal bloom. Now you people would have seen occasionally there can be, you can see red color in the sea. That is because of the presence of phycoerythrin. Okay, this will be more. Okay, there will be pigments. Phycoerythrin will be more in number, which will cause red sea. It will look like red sea. Okay. Yes, what is happening? Yes, today there will be class about biomolecules. I told it in the starting of the class. Okay. Yes, fine. Let's start mycoplasma. Okay, mycoplasma. Now, mycoplasma is the most asked question. Why? Because of so many things, especially this. It is the smallest living prokaryote or the smallest living cell that we know. This is the reason why mycoplasma is asked, every time there will be one or the other question. Okay, one or the other question. That is why I told you cyanobacteria and mycoplasma have some exceptional behaviors because of which they will be asked continuously. Okay, so please do make a note. Okay, please do make a note. Okay, students, look here. They lack cell wall. Now, because they lack cell wall, there are high chances that they become pathogenic, right? Now, penicillin attacks on the cell wall. Now, cell wall is only not there. What will penicillin attack on, right? Penicillium has to attack on something. Now, cell wall only is absent. So, that means what? It becomes more pathogenic. I am telling only about one of it. There are other streptococcus. Those will correctly affect. Okay. Gorniolex you are going to study under dinoflagellates. Okay, do not confuse it with your phycoerythrin in mycoplasma. Okay, most of you might have that doubt. Okay, but this pigment is specifically present in cyanobacteria. Gorniolex you will study it under dinoflagellates. Okay, those are red dinoflagellates. They are not because of just pigments. Got it? Okay, so because of the absence of cell wall, there are high chances that they become extremely pathogenic. Okay. Yes. And then pleomorphic in nature. Students, pleomorphic in nature means they can change their size and shape. Whenever they think. Okay, whenever they want, they will keep changing their size and shape according to their wish. They will change size and shape, and that is called as pleomorphic. Okay, pleomorphic. Just write this down in your textbook. Write pleomorphic and write because of continuous change in size and shape. Some places they even call them jokers of the kingdom. okay, because they keep, jokers keep changing their, colors, right? They keep changing how their moods, everything. So, see, similar to that, we call mycoplasma also that. That is because they are able to change their size and shape whenever they feel so, okay? Now, can you people do that? Whenever you want to, don't feel like studying, you want to become invisible. I don't want to be there. Can you do that? You can't, right? You can't, right? So, that is a speciality of mycoplasma, okay? Speciality of mycoplasma. Did you understand so far? Mycoplasma, you have very less things to remember. It is the smallest prokaryote that is known to us, okay? And they are pleomorphic in nature and they have absence of cell wall, okay? Absence of cell wall. Because of absence of cell wall, there are chances that, penicillium, some of the antibiotics will not be able to attack, okay? Some of them will not be able to attack, okay? Yes. Let's continue. Okay. Now, reproduction. Okay, reproduction, if I have to talk about bacteria. Reproduction in bacteria, all of you know, it is by binary fission. Whenever the condition is favorable, they will do it by binary fission. Got it? Whenever the condition is favorable, they will do it by binary fission. Now, when the condition becomes unfavorable, whenever the condition becomes unfavorable, what is going to happen? They will start producing spores. Got it? They'll produce spores, they'll produce conidia. Conidia are nothing but exogenous spores. Okay? Okay? See, even, what do you tell? Even though the condition is unfavorable, spores will be formed. Spores will be formed. Okay? Got it? That is because they have the ability to withstand any kind of condition. That is why spore formation is always done when the conditions are completely unfavorable. Okay? Got it? So, this point you understood? Reproduction is by binary fission. I don't think anyone will have doubt. Now, when the condition is unfavorable, they will produce by spores or conidia. Sexual reproduction is completely absent. Sexual reproduction is completely absent. Okay? But since sexual reproduction is absent, does that mean that bacteria cannot have variation? Bacteria has never, developed from the stage it was invented or from the stage it was, found. There is no variation at all. Can you say that? So, in your NCRT, there is a statement called sort of sexual reproduction. Okay? There is a statement. Can you all open your textbook? If you find it, tell me. If you found this word or is this statement, sort of sexual reproduction. Tell me if did you find this line in your NCRT? Yes, no. What happened? Where's the energy? So many of you are there. No likes only. No likes only to the session. What is this, students? You should show your energy by liking the session and answering, right? Quickly go like the session. Yes, good. If you have liked, do this. Okay, so fine. Okay, now sort of sexual reproduction. Did you find the sentence in your NCRT? Yes or no? Yes or no? Yes or no? Okay, so sort of sexual reproduction is mainly by 3 ways. Okay, first one is called transformation. Transformation. Okay, next one is called transduction. Okay, last one is called conjugation. Okay, that means sort of sexual reproduction is done by these 3 methods. Now, you don't have to study in depth about any of this, but transformation you have studied in your Griffith's experiment where you saw S strain, right? From S strain, a virulent nature had been transferred or transformed to R strain, and we call this as transforming principle, right? Transforming principle. Griffith gave the name transforming principle, right? This is what transformation means. Got it? Transformation mean. Got it? Okay, some of you are not able to find that sentence. It is there under reproduction, students. Above mycoplasma, there is a paragraph, reproduction. So, in that it is a sort of sexual reproduction. Okay. So, transformation, transduction, conjugation, you don't have to get into depth. This is basically using a virus. We try to infect a bacteria. This is again using variants. Okay. You don't have to get into the depth. So, I am just telling you, but transformation you already know, it means transforming principle. Okay. By this method, by all these three methods, bacteria was able to undergo variation, right? It was able to undergo variation. Fine. So far understood? Yes? Okay. Next. This is, I've put it here for those who are not able to find it. Okay, look here. They have used the statement. They've used the statement. Sort of sexual reproduction. Okay. Sort of sexual reproduction. This is what they mean. Understood? I've put the paragraph. So, here they have told only about fission, unfavorable condition they will form spores, and sort of reproduction they haven't explained, but they have told here, they have told DNA transfer. Okay, DNA transfer. Clear? Clear? Now, we have completed kingdom Monera. We have completed kingdom Monera. So, I will be asking you few questions. Okay? Quickly be ready in the chat box and tell me if you are ready. I will ask you few questions. Ready? A rapid fire. Okay. Want to draw fire. Okay, so rapid fire. Yes, ready? Okay. Okay, put fire symbol if you're ready. If you're ready, put a fire symbol for rapid fire if you're ready. I want quick answers, okay? Let me see. I'm going to note down your names. Okay, ready? Fine. First question. Okay, first question. Shall we start? Okay. Pleomorphic nature is shown by whom in kingdom Monera? Pleomorphic nature is shown by which bacteria? Pleomorphic nature. Yes, great. Pleomorphic nature, which bacteria? Great, wow, good. I like the response. Yes, others, what happened? So many of you are there. What happened? Yes, go on. Okay. So, pleomorphic nature is shown by mycoplasma, right? Okay, mycoplasma. Even though they are small, they are going to change the shape in the size only. They are not going to become like big, small, not like that. Small variations, small variations. Okay, much hasn't been actually figured out about why they're doing it and all that. Later, if they figure it out, we will definitely tell it to you. Okay, pleomorphic nature. Next question. Okay, I'm going to back. Okay, rod-shaped bacteria. Rod-shaped bacteria is called. Rod-shaped bacteria is called. Rod-shaped bacteria. Quickly tell me. Great. Students, why don't you keep your names that I can read easily? It is so difficult that your biological, scientific names, you people have kept your, what to tell, YouTube names. So difficult for me to read. I want to congratulate who told it first. But all the names are very fancy that I can't read. Okay. So, very good. Bacillus is the answer. Okay. Next question. Gas vacuole. Tell me one function of gas vacuole. Gas vacuole, one function. Function of gas vacuole. Function of gas vacuole. Okay, buoyancy. Others, any more answers? Any more answers? All of you, buoyancy? Okay, quickly, tell me. All of you agree with buoyancy? Yeah, buoyancy that will allow them to get optimal sunlight, right? Yeah. Chemosynthetic autotrophism is done with the help of which cell? Chemosynthetic, okay, chemosynthetic autotrophism is done with the help of which cell? Which cell? Cell. Yes, optimal sunlight. One person has told it differently. Okay, Priyanka has told it first. So, this is a heterocyst, right? Heterocyst is a specialized cells that helps for chemosynthetic. Gopika, you have to type the whole thing. What is this? He and dot 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 dot. This is not fill in the blanks. You have to only fill. Cyanobacteria, no. Heterocyst. You people are in so much hurry. Some have typed hetero, some have typed he, she, cyst. Now I have to sit and fill, no? Okay, fine. But I like the energy. You want to be the first, right? You want to be the first. So you're like typing half so that at least ma'am calls my name. Fine. Chemosynthetic. Last question of your rapid fire. Actually, rapid fire should be more quick, but I'm giving you time to type. Okay. Let me think. Okay. Tell me two pigments that are present. Two pigments that are present in cyanobacteria. Two pigments present in cyanobacteria. Two pigments that are present in cyanobacteria. If I ask you specialized cell that will help for nitrogen fixation, if you tell me the heading, no one is going to give you marks, right? You have to tell it correctly. Okay. Chlorophyll B. Chlorophyll B, I never told about chlorophyll B. I told only chlorophyll A. Most of you have typed A and B. Students, chlorophyll A, phycoerythrin, phycocyanin, right? Good job. So, most of you have got it right. So, pigments are chlorophyll A and phycoerythrin or whatever, okay? And the phycocyanin, phycoerythrin. Got it? Okay, rapid fire. Good job. Okay, next, let's start with protista, right? We will start with protista. Okay, so kingdom protista. Now, when I talk about kingdom protista, no, kingdom protista is actually a link between animal, plant and fungi. Do you get it? Get it? Yes, we finished kingdom monera. Over. It's over. We have been taking from last class. How long you want kingdom monera? Okay. Now, kingdom protista, you have to understand, it is a link between plant, animal, okay, plant, animal and fungi, okay. So, that is why it is a kingdom that is really messed up, because we have so many, types coming under that, you will only be confused, okay. So, to break down your confusion, we will just separate it. We will separate who is belonging to plant, who is having the, behavior like a plant, who have a behavior like animal, who have behavior like fungi, okay. Okay, clear? Fine. So, they are unicellular eukaryotes, okay, we know that. That is why we brought about this class. Okay, I'll ask you one more rapid fire question. Let me tell. Who brought protista term first? In the classification, who was the first biologist who brought up protista? Let me see how many of you have revised the previous portions. Tell me, who was the biologist, scientist who came up with it? Okay, one answer has come. Others? Whittaker? Whittaker came with 5 kingdom classification. Okay. 3 kingdom classification. Protista. Who was it? Who was it? Who brought about the classification? Tell me quickly. Those who are new. Yes. Look here. Correct. Ernest Haeckel came with the first protista. So, this is how questions will be asked in your exam. Okay. Which was the scientist who came up with protista terminology? Got it? Okay. Now, yes, here. Okay, fine. So, they are unicellular eukaryotes, fine. Their cell wall may be present in some, may be absent in some, okay. They may be present in some, absent in some, okay. They have presence of flagella, pseudopodia, cilia. That means they have multiple medium for locomotion. Okay, they have multiple medium for locomotion. They have pseudopodia, cilia, flagella. Okay, nuclear membrane is completely present. Nuclear membrane is completely present. They are primarily aquatic and they have ATS ribosome. Because they are eukaryotes, they will have ATS ribosome. Okay. You can go back to my other session and take the screenshot. Okay. Now, fine. Look here. I'm going to make it easy for you. I'm going to make it easy for you. Your protista, no? Your protista. Minute. Okay. Now, in your protista, there are going to be, okay, 3 categories that we are going to make. Because they are a link between animal, plant and fungi, we will separate them. We will separate them. Students, this is the only way for you to study. Otherwise, you are going to find it really difficult. Okay. So, your protista, we will have one called as plant-like. We'll see which are the ones that behave plant-like under protista. Then we will have fungi-like, okay, fungi-like. Then we will have animal-like. Can you all write it down? Can you all write it down? Yes? Ernest Haeckel. Ernest Haeckel. Okay, plant-like. Plant-like we have cryophytes. Okay, we have cryophytes. We have dinoflagellates. Okay, we have dinoflagellates. We have euglena. Okay, euglena. We have here slime mold. Okay, slime mold. Here, animal like, we have protozoans. Okay, students, these are the, no, cryophytes, correct, no? What happened? Okay, so plant like, we have cryophytes, dinoflagellates, euglenoids. Under fungi, we have slime molds. Under animal like, we have protozoans. Now, I'll tell you why we have considered them. Okay, why we have considered them. See, in this, no, most of them will have cellulosic plate, like the cell wall will be made of cellulose. Some of will be capable of doing photosynthesis. Okay, okay. Here, slime mold, they are mainly heterotrophic. That means they feed, they're saprophytic. Under heterotrophic, they are saprophytic. They feed on dead and decayed. Here, protozoans, when we talk about, they have locomotion. Okay, here under protozoans, you will see the most locomotion. You can see it by cilia, you can see it by pseudopodia. Okay, you can also see it by flagella. I'm not saying others do not have flagella, but here they will do proper locomotion. Okay, so this is animal like. Now, once you clear this, no, you will understand. You will understand, okay, what should I write for this? What should I write for this? Even in case you get confused, you can look at this box and understand. Clear? Clear? Okay. Now, look here, your protozoans are so many types of 5 types. Okay, cryophytes, dinoflagellates, euglenoids, slime mold and protozoans. Under protozoans, we have amoeboid, ciliated, flagellated and sporozoans. Okay, make a note, all of you. All of you make a note. Make a note, all of you. Okay, fine. Did? So, protista, we have starting with cryophytes. Okay. Once you are done, let me know in the chat box if you have made a note. If you have made a note quickly. Yes. Okay. Okay. Fine. So, let's start with the first ever one, that is your cryophytes. Okay. Under cryophytes, students, under cryophytes, okay, under cryophytes we have desmids. Desmids, that is your first example. Your NCERT starts with that, telling that cryophytes have desmids and diatoms. Desmids are also called as golden algae. Okay, golden algae. Fine. This golden algae and diatoms together contribute your cryophytes. Together they make cryophytes. Fine. Now, they are seen both in freshwater and marine water, and flagella is absent for them. Flagella is absent in this. Okay. Then they are floating, that is why they are called as phytoplanktons, because they are floating, they are called as phytoplanktons. Okay, fine. And they have a cell wall, that is, yeah, here when we talk about diatoms, no, you should know that their cell wall is made up of soap box like cell wall. Very nice, no? Now you have a down, okay, down place, and we have a top portion, right? Now it is like a soap box, so down. Now imagine, how do you keep your soap? Okay, this is the plasma membrane, and they have a cell wall which is like an overlapping shells. Okay, which is overlapping shells. So they have overlapping plates, shells. Okay, you can tell them, oh, they are overlapping, and they are in the form of a soap box. Soap box. Right? All of you have seen a soap box? Yes? No? Yes, very good. Okay, most of you are telling what is it made up of also. Very good. But have you all seen a soap box? Normal soap box. Right? So, this soap box is made up of a combination of, cellulose and silica. Okay, it is made up of a combination of cellulose and silica. Now, what is going to happen? Okay, what is going to happen? Now, look here. This is your soap box structure. Okay, I am drawing it big enough, right? We have our plasma membrane inside this. Okay, now this is a combination of cellulose and silica. Okay, and this silica, no, this silica is a very rigid hard substance. Okay, so this silica will not be easily destroyed. Okay, it cannot be easily destroyed. So what is going to happen? This diatoms, once this diatoms die, okay, once this diatom die, okay, all the cell wall will go and settle on the seabed. Okay, will go and settle on the seabed. Okay, okay, will go now. Think of this. Okay, all your diatoms, okay, have gone and settled, right? They have gone and settled on the seabed, and they will form another layer. Okay, they will form one whole layer of silica. Okay, one full layer of silica, and that is called your diatomaceous earth. Okay, diatomaceous earth. Got it? Got it? Diatomaceous earth. Clear? Clear so far? Now here you can look, this whole silica has come and settled on the seabed, and they have formed a completely new bed for under the sea. Okay, that is called your diatomaceous earth. Now this diatomaceous earth is rich in silica. All of you agree? Rich in silica. They are very, very rich in silica. Okay. Now what is going to happen? This First, okay, first the purpose was not known. Okay, purpose was not known. Later, humans started scraping this out. They started removing this diatomaceous earth and they started using it for other purposes like polishing, for filtering, okay, for cleaning, okay. They started taking diatomaceous earth for other things like polishing, cleaning. Got it? Got it so far? Okay, yeah, filtration. It was even used for filtration. So, under cleaning itself, you can add filtration. Okay. So, this cell wall ended up forming a completely diatomaceous earth, a complete seabed full of silica cell wall, which were completely different in color. Okay. Okay. Fine. Let's continue. Let's see what your NCERT have to say, right? So, your cryophytes are mainly made up of diatoms and golden algae. Students, I am also doing NCERT reading, right? After every topic, I am taking the snips of the NCERT and explaining it to you. So, your work is completely reduced. Now, your only job is to study, right? Your only job is to sit and study. So, make sure that by the end of the class, you have a complete notes of your NCERT, because whatever I am making as a notes and giving you is more than enough. Upon that, I am also referring your NCERT to check if any points are missed. Clear? So, your actually 3/4 work is over. Now, your main job is to sit and study. Got it? Got it? Tell me, how many of you are studying every day now? How many of you are revising every day or studying whatever was done last class? Tell quickly. No one. No one is studying. No one is studying. Wow. No one is responding. Okay. You're all studying. Great. Okay. Fine. Be consistent. Same thing. Be consistent. Okay. No problem. You're a dropper. You're a new person, like you're newly joined or you're planning to write. For everyone, this is helpful. Okay. So, if you are a dropper and you really want to crack NEET, you should join a batch and have proper guidance, okay, proper guidance from educators, so that you will have a track of where you are doing or what you are doing. Otherwise, there are high chances, when no one is watching you, you will not study that well, okay. So, if you want to join any batch, download our app and join any batches that you want. Even for special classes, special classes are completely free. You can still use my code Gopika and you can join the batches and you will get the best guidance and also exclusive discount. Okay, clear? Fine. Okay, now look here, this is your NCERT. Fine, they are found in freshwater, marine environment, they are microscopic and they float passively in water. That is why they are called as plankton. Now, most of them are photosynthetic, okay. In diatoms, the cell walls are made up of two thin overlapping shells, okay, two thin overlapping shells, and which fit together as a soap box, okay, a soap box, fine. The walls are embedded with silica and thus the walls are indestructible. This is why I told you silica is very hard and you cannot destroy it easily, right? Fine. Now, the diatoms have left behind a large amount of cell wall deposit in their habitat and this leads to diatomaceous earth. Okay. Students, again a very common question from NEET. Okay, very common question. So, do understand diatomaceous earth. Now, that is used for polishing, filtration of oils and syrups. And diatoms are the chief producers of the ocean. Now, they can ask you a question, which is the chief producer of the ocean. Okay. Fine, got it? Yes, got it. Gopika, you can join other batches. Okay, it looks like I'm talking to myself. Gopika, you can join other batches, right? You don't have to depend on that. You can join the other batches. Okay, and you can use your own name as a code. How lucky are you, right? You can use your own name as a code to join. Students, I have done MCQs for molecular basis of inheritance in my special class, and it was an amazing session. We had a lot of students and most of them, was very competitive, and it was a really nice session. So if you haven't still gone and seen, please go check molecular basis of inheritance. MCQs have been done, not PYQs, okay, not past years. I have done new questions, okay. Fine. Let's go into dinoflagellates. Students, from the name, dinoflagellates, what do you understand? What is there? What is there? Dinoflagellates. Flagella is there. Flagella is there. Right? Dinoflagellates, the word itself says that they have presence of flagella. Okay, they are marine. They have two flagellas. Now, two flagellas means they have two types of flagella, which is, one is longitudinal, okay, longitudinally formed. Another one is transversely formed. Okay, they have grooves. From the grooves, longitudinal arises, transverse arises, and they will form a perpendicular structure. Perpendicular structure. Got it? Okay, this longitudinal and transverse will form a perpendicular structure, and that is why they have two flagellas. Okay, now look here, students, this whole through is protozoans, no? Cell wall is one important thing. Because everything, say your cryophytes, dinoflagellates, slime mold, your euglenoids, all of their cell wall, no, has one or the other exceptions. That is why this is a favorite question. This protozoan, this protista itself is a favorite question for all NEET. Okay, so people who are taking it too lightly, do not take it lightly. You might even lose like 1 or 2 marks because you are careless. Okay, so do listen to all the exceptions. Fine. Now, they will be perpendicular to each other. They have cell wall that is made up of cellulosic plate, which is made up of pectose. Okay, they have a very rigid, okay, they have a very rigid plate like structure that is called the pectose. Pectose, clear? This rigid layer is called as pectose, and they are made up of cellulose. Yes. Okay, so your NCERT says about, here is where you will be coming across Goniolax. Okay, now these organisms are mostly marine and photosynthetic, and they appear in yellow, green, brown, blue, red. That means so many pigments are present. Got it? They can be seen in many colors. Got it? Yes. Now the cell wall has a stiff cellulose plate, okay, on the outer surface, and most of them have two flagella. Okay, most of them have two flagella, where one is longitudinally arranged, the other one is transversely arranged. Okay, and here red dinoflagellate example is Goniolax. Okay, students, this Goniolax, what does it do? No, it will continuously multiply. Okay, continuously multiply, and what happens when the high tide or low tide is hitting, no, you can see that area is completely red. Completely red. It will look like, as if someone has opened a bottle of, your food color or red paint into the sea. There will be some regions which are blue, some regions which are red. So this causes red tide. Red tide like appearance. That is because this Goniolax continuously does rapid multiplication. They will be so much in number that will end up forming a red color pattern. Okay, now you might be thinking, oh, such a beautiful thing, it will be showing the sea red in color. Now the beauty is not, beauty cannot, like, you cannot judge a book by its cover. No, same thing only. Your Goniolax releases toxins. Now what happens, no, this is similar to your how your algal bloom will happen. Because they are rapidly multiplying, they are going to release a toxin. That toxin will get affected by other aquatic organisms, okay, like your oysters. Okay, now this other marine organisms are going to absorb this toxins released by Goniolax, and when we consume it, when we humans consume it, we end up having serious infections. Maybe it can reach to such a fatal condition where you could die. Okay, so understood, not everything that you see beautiful will be, what to say, not everything beautiful is I forgot, students, don't judge a book by its cover. Okay, that's all. Okay, red tides, toxins released by such large numbers may even kill other marine animals such as fishes. Okay, fishes? Okay. You can first download the app, join my special class. I'll tell you the details. Okay. Gopika, you can download the special class through the description box and then I will tell you the details. Okay. Fine. Okay. Now, students, this is the image of the Red Sea. Can you see this red color tides here? Can you see how this Goniolics have completely rapid multiplication has done? Okay. Okay. And they have formed this red tides. Can you see? How can you see the difference? There is like greenish blue and there are some regions which are red. Correct? It looks beautiful, but then they are releasing a toxin that is killing other aquatic organisms. So, can we call it beautiful? It is harming other organisms, no? Yes, looks very beautiful. Correct. Looks very beautiful, but what to do? Okay. Next one is your euglenoids. Okay. Okay. Euglenoids. Now, this euglenoids, cell wall is absent. Okay. Because they do not have cell wall, they have a proteinaceous layer called pellicle. Okay, they have a proteinaceous layer called pellicle. Okay, this proteinaceous layer is very flexible. Okay, very flexible. Okay, and because they do not have cell wall, this one pellicle will do the job of protection, everything. Right, protection, everything. Now, they have two flagella. So, students, there are high chances that you get confused with dinoflagellates flagella and euglenoids flagella. Understand that dinoflagellates flagella is, one is longitudinal, one is transverse. Okay. Euglenoid, one is long. Look here, one is long, another one is short. Okay, one is long, another one is short. Got it? Got it? Okay. The dinoflagellates also have two flagella. Euglenoids also have two flagella, but one is long, one is short. Okay. One is longitudinal, one is transverse. So, do not get confused. Okay. Do not get confused. Fine. They have contractile vacuole that will help them for osmoregulation, right? Balance. They will help them with osmoregulation. In the absence of light, they can feed heterotrophically. Now, Euglena has many specialized characters. Euglena can do autotrophic, can be autotrophic. That means they can make their own food. Okay. But when light is not there, they can feed on smaller organisms. Got it? So, they are heterotrophic. That means what? Opportunist. That means whenever opportunity come, it will become heterotrophic. Whenever opportunity is not there, they will, whenever there is enough sunlight, everything, they will do photosynthesis or they will do autotrophic behavior, make their own food. Got it? Now, when the condition is unfavorable or when there is no enough light, they will do heterotrophic behavior. That means they will feed on smaller organisms. Got it? Yes. Now, they have pyrenoids that allow them to survive. This light, this has got cut. It basically means they have pyrenoids, that is a type of carbohydrates, okay, that will help them to survive. It is kind of like a starch, okay, it is kind of like a form of starch, okay, we can call it as a form of starch. They will allow them to survive during the condition when they don't have light, like wherever, like, if they are like not in a place where there is no enough light, this pyrenoids will help them to survive. The starch, this pyrenoids, they will help them to survive their portion or that period of time where their light is not there. Got it? Okay. Fine. Let's see what your NCERT says again. Now, majority of them are freshwater organisms and found in stagnant water, okay, and they do not have a cell wall, so they have pellicle, which will make their body flexible. I told you. Now, they have two flagellas, one is short and long, and though they are photosynthetic in the presence of sunlight, when they do not have sunlight, they will do heterotrophic nature. Okay, they will start feeding on other organisms. So far good, so far good. Tell me in the chat box, so far, did you understand? Is the chapter difficult? Isn't it easy? Tell me quickly in the chat box, is it easy or you're still finding it difficult? What is yes, ma'am? Tell easy, difficult. Okay, very easy. Fine. Easy. Okay. Okay. Yeah. So they, in the presence of sunlight, they will do photosynthesis. Absence of sunlight, they'll become heterotrophic. That is, they will feed on smaller organisms. Now, the pigment. Okay, students, here Euglena has a pigment called chlorophyll. Where do I write? Yeah, I'll write here. Chlorophyll B, that is only present in higher plants. Okay, that is only present in higher plants. Okay, this is why they have used the word that the pigments of euglenoids are identical to those present in higher plants. They have chlorophyll A, they have xanthophyll, they have carotenoids. They also have chlorophyll B, which is only present in higher plants. So, Euglena has the pigment that is present in higher plants. Got it? Got it? I hope you don't have any doubts from NCERT, because I am going line by line. Okay? If you are with me, then definitely by the end of the sessions, you will be thorough with Kingdom Protista. Okay? Great. Now, next one, slime mold. Okay, now tell me, let me see if you are listening to my class or not. Tell me, slime mold is showing what kind of characteristic? Is it animal, plant, fungi? Slime mold is showing what kind of characteristic? Animal, plant, fungi? Quickly, I'm waiting. Okay. Is it going to behave like a fungi, animal? Okay, again, good. One person has answered. Fungi-like. Yes, fungi-like. Okay. It is told fungi-like because it is going to do saprophytic. That means it feeds on dead and decayed, and they also form spores. Okay. Fine. Now look here. They are heterotrophic. Especially they are very much saprophytic. Okay. Saprophytic. They live on organic matters. Okay. Saprophytic, and absence of flagella. They do not have flagella. Now here, they form an aggregate. Can you see? This is the aggregate. Okay. They form an aggregate called plasmodium. Plasmodium. Okay. This is an aggregate actually. That means, aggregate means what? Your aggregate, your marks aggregate. That means totaling of everything or your average of everything. Same way, this is a cluster. Okay. This aggregate, it's called plasmodium, and it will be formed during favorable condition. This is what you have to remember, students. Plasmodium will be formed only when the condition is favorable. When the condition is favorable. Okay. Now, what are they going to do? They are very smart. Okay. They are very smart. Now, condition has become favorable. They will form plasmodium. Now, condition will become unfavorable. Correct? No? Condition will be always favorable. How many of you have always made a timetable and followed it? How many of you have made a timetable and always followed it without, 1 minute here and there? Not possible, right? Condition will always not be favorable. Sometimes condition can become unfavorable. Now, same way, slime mold knew that the condition could become unfavorable. So, what it will do on the plasmodium, right? Plasmodium, now this is your plasmodium. On the plasmodium, it is going to have fruiting bodies. Okay, it will have extra fruiting bodies like this, and inside this fruiting bodies, they are going to be spores. Okay, on this fruiting body, on the sides, they are going to form spores. Okay, got it? Got it? So, during unfavorable condition, fruiting bodies are formed on the plasmodium that produces spores, and this process can be called fructication. Okay, fructication. Now, look here, they can withstand extreme condition. Who can withstand extreme condition? Spores can withstand extreme condition, right? Spores can withstand extreme condition. That is why they are being formed during unfavorable condition. Okay. So, favorable conditions, slime mold will form plasmodium, which is an aggregate. When conditions become really unfavorable, on the plasmodium, tip of the plasmodium, they will form fruiting bodies. Inside the fruiting bodies, spores will be there. Now, the spores will, this fruiting body will rupture. The spores will be scattered with the help of air currents, right? This fruiting body will be Okay, they will be scattered with the help of air currents. Okay, now this will get scattered with the help of air. Now they will scatter and the multiplication will continue. Okay, multiplication will continue. Students, I'm a Mallu. I'm from Kerala. I'm not, I am a Malayali, but I'm born and brought up in Bangalore. Okay, more than this, I cannot tell. I kept introducing myself in every class. Okay, those who are new to my class, I am a South Indian. I talk Malayalam. Okay, last class I have even spoken Malayalam because the students were not allowing. Okay, now slime molds are saprophytic protists. The body moves along the decaying twigs, okay, decaying twigs and leaves, engulfing organic matter. So, under suitable condition, they form an aggregation called plasmodium. Okay, they form an aggregation called plasmodium. Fine. Okay, and which may grow. So, which may grow, what happens? The spread over several feet. So, during unfavorable condition, this plasmodium will differentiate. Plasmodium will differentiate and this spores will get released. Okay, and the spores possesses true walls. Students, even if slime mold does not have a proper wall, the spores will form proper wall. Why? Because spores have to withstand unfavorable condition, right? Right. So, spores will have true walls and they can withstand any kind of condition. Even for many years, they will sleep like this only. They will not get affected at all. If it's raining, if it's thunder, it is, it's very hot, very cold condition also, the spores will happily be there. Because they are made for that. They are made to survive unfavorable condition. Okay, got it? Now the spores are dispersed by air currents. So we have reached up to slime mold. Okay, now the last protista that we have to study is your protozoan, right? Your protozoan. Under protozoan, okay, under protozoan, you have, okay, under your protozoan, okay, you have amoeboid, okay, amoeboid, you have ciliated, you have, right, flagellated, okay, and the last you have sporozoans. Now students, half of the answer is in the name only. Half of the answer is in the name only. Okay. Why I'll tell you half of the answer is in the name. Okay. Cell wall, I'll tell you why the name is. Now cell wall is absent in protozoan. They are heterotrophs. Okay. These two characteristics. Now tell me these two characteristics, where have you seen? Have you seen it in plants, fungi or animals? This absence of cell wall and being heterotrophic, where have you seen? Have you seen it in plants, animals, fungi? Okay, tell me quickly. Cell wall is absent. Okay, cell wall is absent and they are heterotrophic. That means they depend on other organism for food, right? Now tell me, this nature is seen in? Yes, very good. It is seen in animals, right? It is seen in animals, right? So, this two characteristic is a big yes that okay, this should have been belong to animal. That is why we say that protista is a combination or a link between animal, plants and fungi. Animal, plants and fungi. Okay. So, cell wall is absent. This two characteristic possess like mainly shown in animals. Now, let's get into the first protozoan, amoeboid protozoan. Okay. Tell me the most specialized characteristic of amoeba. Most specialized characteristic. Like every time you talk about amoeba, this word will be there. Tell me. Quick. Which is the most specialized character of amoeba? Every time you study about amoeba, you will have to study this word. No matter what, you will have to study this word. Quickly tell me. Yes, very good. Pseudopodia, right? Pseudopodia. You would have never studied amoeba without studying pseudopodia. Same students, amoeboid protozoans also have pseudopodia that will help them like a false feet, right? That will help them for locomotion and engulfing food, okay? To take the food in and for movement, the pseudopodia will help. So amoeboid protozoan, you will never get confused. You can always think about, okay, amoeba means pseudopodia, right? Fine. They are seen in freshwater, seawater and moist areas. So even if they say terrestrial, okay, it means that it is present in moist areas, okay. And then sexual reproduction is absent. They are free living and particle feeders, okay. So whatever the pseudopodia is going to engulf, that is what these people are going to eat, right? They are going to feed on that. That is why we call them as particle feeders. They are not like, they will not engulf a big organism that is bigger than them. They are going to engulf small, tiny things, okay. Whatever they can break it down. Got it? So amoeboid protozoan, you have to talk about pseudopodia. Okay, pseudopodia. Right. Now, ciliated protozoan. Now, ciliated protozoan, you can look here, paramecium. You remember paramecium? Okay. You always think about the sole, okay, sole of the shoe, right? So, this paramecium, they have small cilia, okay, small cilia like hair like structure. That is why this is called ciliated protozoan. They are free living. They have cilia, which is basically hair like structure, okay, hair like structure, okay, and that hair like structure is the reason why they help for locomotion, okay, why they help for locomotion and taking food inside, okay. Got it? Yes. They are obligate parasite. They are obligate parasite, sometimes opportunists. That means they will do parasitism and predatorship. Okay, they can do both. They can be predators or parasites. Depending on the condition, they will change, students. They are chameleon nature. They will be like, okay, today I want to be a parasite because a host is there, I will become parasite. Okay, sometime I want to be a predator, I will become a predator. So, they are obligate parasite and sometimes predators. Now, here, look here, they have two types of nucleus. Okay, why I am talking about nucleus is one specialized character of them. This is called micronucleus, okay, or you can call them as generative nucleus, right? Look here, generative nucleus, okay. Red is visible, okay. They can be called as generative, okay, nucleus, okay. And this, the bigger one, no, the bigger one that you are seeing is your vegetative nucleus, okay, is your vegetative nucleus or macronucleus. You can also call it vegetative. Now only mark it in your textbook, students. Mark it in your textbook. We have micronucleus and macronucleus, okay. Okay, got it? We have micronucleus and macronucleus. Macronucleus can also be called as vegetative nucleus, that will basically help in all cell regulation, okay, metabolic activities. And then we have micronucleus, that is the generative nucleus. Generative nucleus will be helping, okay, generative nucleus will be helping with what to say, the reproductive parts, okay, for the generative, reproductive, got it? With that part, this micronucleus will be helping. Got it? Did all of you make a note? I don't think any of you made a note. If you made a note, put a thumbs up in the chat box. If you made a note, put a thumbs up in the chat box. Tell me. If you made a note so far, micronucleus, macronucleus, vegetative nucleus, it will basically do, you know, cell regulation, okay. And this your generative nucleus will do all the reproduction or generative cell reproduction, all the reproductive activities will be done by it, okay. Yes, good, okay. Few of you have written, few of you have not written. Students, when you are in the class, take maximum utilization of the class. Don't go again, watch the video and you will sleep, okay. So, they have two types of nucleus. Now, look here. This is your contractile vacuole, okay, this is your contractile vacuole, which looks like a sun, flower, whatever you want to say it, okay, contractile vacuole. This is your oral groove. Now, oral groove is from where, okay, all this particles will be taken in, okay. You can also call this gullet, okay, G-U-L-E-T, gullet, okay. Got it? Now, look here, contractile vacuole, oral groove, the E went off, okay, oral groove, fine. This micronucleus, macronucleus, and we have contractile vacuole, okay, got it? This is your euglena, okay, yes, here only this oral groove will lead to an opening called your gullet, and that will allow euglena to absorb all the nutrients that is required, okay. Sorry, paramecium to absorb all the nutrients that is required, okay, fine. Last one, I mean not last one, flagellated protozoan. After that you have one more, okay. Flagellated protozoans, they are free living, they have flagella that helps in locomotion, they are parasitic, they are pathogenic, okay. Trypanosoma is given in yours, that is sleeping sickness, okay, sleeping sickness, fine, okay. Here nothing much to study, students. Here only you have to know is about this one. Example you have to know, and you have to know that they are free living and they have flagella. From the name itself, you know, ciliated had cilia, amoeboid had pseudopodia, flagellated will have flagella. Easy, no? With the names itself, half of it is figured out. Got it? Okay. Now, example is important. They are very pathogenic. Okay, they are very pathogenic. They can cause maximum infections in human. So, trypanosoma causes sleeping sickness. Okay, sleeping sickness. Fine. Why are you all so much distracted in the chat box? Students, focus. What is this? I'll ask you a question now. I'll ask you rapid fire. If you people don't answer no, then I'll know how much you people are listening. Okay, sporozoans. Sporozoans, they are endoparasitic. Students, sporozoans, there's only two sentences that is given in your textbook. First of all the sporozoans will have a spore-like stage. From the name itself you understood, sporozoans, spore-like stage. Got it? And they are very infectious because, you know, because spores are very, what to tell, they are very resistant, no? They are very resistant to most of the extreme habitat. They cannot be killed easily. Okay, they cannot be killed easily. So, that is why we call them, they are very, very infectious. Okay, and the spore-like stage which makes them highly pathogenic. Now, plasmodium causes malaria. All of you know the plasmodium. Students, you studied two plasmodiums in this class. One plasmodium was an aggregate, okay, aggregate formed by slime mold, which was completely different. Here, this plasmodium is your disease causing plasmodium. Got it? That causes malaria. So, do not get confused. Don't write aggregate, plasmodium, malaria. Khichdi you don't make. Okay? So, they have spore-like stage which makes them highly pathogenic, and plasmodium is the example which causes malaria. Clear? Clear? So, like that we have come to the end of kingdom Protista. Kingdom Protista. Got it? We have come to an end of kingdom Protista, kingdom Monera. Now, we have fungi, plantae and animalia. Got it? Got it? Now, shall I ask you questions? Shall I ask you questions? Yes, ready? Rapid fire? Rapid fire round ready? Okay, rapid fire round 2 this is, right? Students, this time you have to be quick, okay? First come, first serve. Whoever answers first will get, only their answer will be accepted, okay? First come, first serve. Yes. Put a fire symbol if you are ready for rapid fire. So, this is going to be always there in my class. After every topic, maybe we will have a 5 question round which is called the rapid fire round. So, if you are ready, put a fire symbol. Okay, let's make it like one seal for Gopika ma'am's class. Got it? Anytime rapid fire you want, you would put fire symbol in the chat box. Why I can't see chat? No symbols I can see. Why? No one is ready. Yeah, tell me. Very good. Yes, great. Others also ready, even if you joined late, just give it a try. Just give it a try. It's okay. It's okay. Okay? I'm not going to mark you, but I will check how many of you are in the class. How many of you are in the class? Okay. Fine. first question, just because we finished it, I have it in my head fresh. So, macronucleus is also called what in paramecium? Macronucleus is also called dash. Yes, macronucleus. First come, first serve, okay? I'm not going to look at your messages if you're waiting to type for another person, okay? Yes, Nandini has answered first, okay? First come, first serve. I've picked her name. So, vegetative nucleus, right? Vegetative nucleus, okay? Fine. Second one. Second question. Ready for second question? Okay. So, after many of you have answered, okay? Yes. Okay, fine. Look here. Students, if you have any kind of doubts, how do I start, what do I do and all that, you have to go to my description box, download the app and join my special class. Follow me on, follow my profile, okay? So, when during the class, I can let you know what could have been done, okay? Fine. I can help you, but now I cannot help you because there's a lot of other students and we are on a theory class. So, any questions that you're finding it difficult to ask any other teacher, feel free to join my special class and ask me, okay? Fine. Yes, fine. Second question, students. Second question. Flagella present in dinoflagellates. Flagella present in dinoflagellates. Types of flagella present in dinoflagellates. Two types. Tell me the types quickly. Tell me the types. Dinoflagellates. Two types of flagella they have. What are the two types? First come, first serve. Quickly type it. Okay. Okay, there's lot of competition. I like it. Very close timings. Okay. Nandini and Subhashni are very close. Okay, we have transverse, right? Transverse and longitudinal. Okay. And one person has told they form a perpendicular structure. Correct. Okay, they are perpendicular flagella. Okay. Third one. Third question. Let me think. Okay. I'm going to ask from everywhere, okay? Sort of reproduction, what is the three techniques that are used in bacteria? Sort of reproduction, what are the three types of sort of reproduction that you studied? Sort of sexual reproduction is of three types. What are they? Sort of sexual reproduction in bacteria are of three types, right? What are they? What are they? Okay, very good. Again, okay, so it's conjugation, transformation, okay, transformation, transduction, right? Transduction. Okay, last rapid fire, no, fourth question. Fourth question. Contractile vacuole. Contractile vacuole, is it present in paramecium? Okay, is it present in paramecium? Ciliated protozoans, do they have contractile vacuole? It's a yes or no question. Just put yes or no. Yes or no. Very good. Many of you have answered transduction, conjugation, no. Okay, Aishwarya's answer is no. Others, do they have contractile vacuole? Yes, yes. Yeah, the answer is yes. I told you, no, they look like sun. You saw just now, they look like sun, right? Like this. Oh no, my sun collapsed, students. Okay, there is sun-like structure. Just now I labeled it. Okay, so contractile vacuole is present. Okay, yes. Okay, last question for today's session. Last question. I have to think. Okay, last question. Red tide is caused in dinoflagellate by which example of a dinoflagellate that causes red tide? Example. Example, tell me example. Dinoflagellate example. Okay, great. I like the response. Okay, I like the response. It is very, very great, right? Gorniolex is the example. Gorniolex is the example. Okay. How many of you have answered? Most of you have answered. Very good, right? Great. It is an example of dinoflagellate, Gorniolex. Okay, Gorniolex. Got it? So, students, finally we have completed. Okay, finally we have completed Monera, Protista. It is definitely a long chapter, but we can do it, right? Who else can do it? We can do it. No, we are strong. We will do it, right? We will sit for how many hours that it takes, and we will complete the chapter, right? Then only I can shift to next chapter. Correct? Correct. So, do not worry, we will start next chapter, fungi. Now, once again, I am telling you people, those who joined late, many of you have asked for biomolecules explanation. I have started a series, I started a special class session in my, in my profile. So, do not forget, go to the description box, click on the username, and you will be directly downloading the learners app, and you can join my classes on biomolecules, where I will be clearing your doubt and teaching you repeatedly. How many ever times you asked out, I am still going to explain it to you. Okay, so you can start biomolecules with me in my special class. Those who don't know anything about special class, it's all in the description box. Okay, you have to follow me to get all my links. Clear? Clear? Everything, all the details down in the description box. Check it and join for biomolecules, because I know it is a difficult chapter. Okay? Clear? Clear? Thank you, students. Thank you so much for joining. It was definitely an amazing session, like all the time. Okay? Do not forget to like, and if you have any doubts so far in the chapter, put it in the comment section, because I will be reading all your comments. Okay, whatever feedbacks you want to tell me, whatever you want to tell me about the class, put it in the comment section. Okay, as soon as this live gets over, put it in the comments. I will definitely read it. Okay, and I am definitely waiting for your feedbacks. And if you have, ma'am, no, this is not the way, ma'am, I want it like this, whatever it is, you can tell it in the comments. Okay, I will be reading the comments, and I will revert back in the next video. Okay, clear? Take care, be safe. Okay, thank you so much for joining. Okay, and do not forget to join my special class, and do not forget to use my code. Okay, see you all.